Okay, good afternoon, everyone. Very nice to be here. I'm Judge Marko-Várga, and I'm going to tell you a little bit about the development that we have with formalin-fixed material. So this is a great asset since pathology department have very, very big collections over decades with these samples. So I'm going to show you some studies on melanoma, and you're well aware, I mean, this is the, one of the most fast progressing metastases, cancer disease of all. And we also have a very high mortality rate that actually we have not really been able to conquer and don't have a very good solution for, even though we have new pipeline for big pharma introducing immunotherapy and other ways of handling it. So here is a little bit of statistics of where we're going. Still going up, even though diagnosis is not the best, and treatment is also, we would like to expect more. Now, that is true both for men and women. You can see here, top scale, you have Australia and New Zealand, and below you have countries like UK and Scandinavia actually is pretty bad. However, if you find the diagnosis very early, you have a very good chance. And as you move into, let's say, stage 2 or so, not so good. So because then you're already under pressure. And then we are just, in principle, in the hospital, we're kind of extending lifespan of patients. But doing so, we have a clinical team. We really work together, multi-center studies around the world. So here's Olivia Esto, Marcel, and Balázs. And also, we work a lot into the clinic as well in surgery. And in surgery, we have a very good established collaboration. So we actually get clinical material. So what you see here is in a hospital where we are just down the corridor and able to process and handle surgically isolated material. So from there, of course, then you have the workflow in the hospital. You go into pathology. And now we built a super big biobank. It's actually here in Hungary in Central Europe. And to the right-hand side on the image, you see where we already started. So this is 650,000 tumor blocks that we enter in. And in the middle, that's actually the whole archive with 6 million. And to the left-hand side, you see how everyday work is done and how they are cataloged. Now, we are addressing actually a build of knowledge in understanding both intra-tumor heterogeneity and inter-tumor heterogeneity. So we are interested to understand, I'll show you a little bit later how we do that, the clone specificity. So how can we identify that? And what about the immune defense? How does that work out together? And we are now moving up to very high numbers of images and patient tumors where we are kind of building knowledge on that. And in the end of the day, of course, many of us in my team is from big pharma originally, now moved over to hospitals and into academia as well. But we are very interested in the mode of drug action and also to understand the mechanisms driving that. But in the end of the day, the most important is actually to go where the problem is, and that is within the tumor tissue. So here is an example. Early on, we started to catalog. We built very big databases where we started to annotate these different regions. So this is like early setting, and if you go from there, you can actually now start to do modeling. So you can find different compartments within the tumor where you can identify different tumor clones. And if you go one step further, you can actually then move from there to have single cell annotation. And in this way, you will have very high numbers. So imagine you get like 600,000 cells identified within one section. And then you move up to 1,000, 10,000, 100,000 of sections from multiple thousand patients. Well, you get my point. And then from there also we are kind of attacking in a single cell mode. So on the right-hand side you can see how we isolated single cells within tissue from activity that we're driving by laser microdissection. And the whole idea here is also to have spatial readout. and to understand what is really happening in compartments within the tissue. Now, building this up, we decided, so Letizia and Lucas have been pushing this now for some time, to use an open source, so it's a QPath, it's available publicly, and then we kind of build our model platform on this platform. So you start with setting it up, getting holistic whole section view, and then as you move forward, you're able to move into the details and then also to build neural network, and from the neural network you're actually able to get to a situation where you have an ability for automated annotation, and that is on single cell level. So, you know, there are not so many pathologists in the world that will be able to help you, so we need computation power in order to manage go forward. And so what we're doing here is that we're using augmentation technology, and I show you a little bit of an example here. So what do? So you have one original image. This is the one to the left, right? And then what you do, you actually make a virtual copy of that. What you do next, then you can play around, so you can make a mirror image, and in this way you're expanding the number of images that is accessible now. You can also rotate it, you can actually change the color of it, and by that you actually also are able to generate from one, you get up to 500 or multiple hundreds of images, and this is how you actually manage, and then you build the model onto that. In this work, we published very early a study. We were the first in melanoma with AI segmentation, and we have a collaboration with NIH in the Cancer Moonshot program that is the biggest right now, and we have published a number of studies here where we together with the NIH have been going forward. I'm very happy. It's a great team and effort, and we were evaluating, and we're just moving into phase 2 of this program. So it's not only US, it's global, worldwide today. Okay, so with this in mind, I show you next study example of where we're going, and this is the MaxMel study. So here you see 4 different type of classes of the melanoma, and the acral melanoma is the most pronounced in Mexico. So this is a study where Diana has taken the lead, and we have 1,000 patients here. And everything I'm showing you now, data, is actually generated in the collaboration done with Covaris technology. So Eric has presented the details of operational of how it's being executed. So here's a workflow that we worked out, going from sectioning, histology, and then going downwards and workup, and then you also need to actually solubilize. So what you want to make a solid sample a liquid, and you want it a fully solubilized liquid. And this has been very efficient and fruitful, and we spent quite some time in getting it right. So, you know, so it's easy to say, yeah, you do like this and do like that. Well, it was more than that. So we were able to get an understanding of the timing, how we should do it, and then, as Eric alluded to, we can do 96 well format in this high-throughput system. And this is very important for us because now we're generating study data that you see here. You can really do deep sequencing, you can quantify the 4 different classes of melanoma, and you can also find key regulating proteins in the respective class of melanoma. I will not go into the detail of one of the targets that we are kind of looking into patenting now. But if you want to also have an understanding of how they relate to one another, you can actually do the acral, that is the main importance for us, and then how also you can then do the analysis and check for the other 3. So this is these are data from 2020, and now we are moving into large scale for the Diana-driven part of the 1,000 patients. And here you see it's actually fresh from the bakery, so we generated these data just before coming here. And this is what you see as a display. So this is the total amount of proteins generated. You kick it up to 6,000, 7,000, 8,000, and then with the database, you're able to crunch even further. And when you compute, what you do is then you go in and you need to also to normalize data so you get an equilibrium, because this is run over a period of time, and you have internal standard, you have controls, and also we have a mixed control that we use. And by doing that, then you can get really reproducible and very strong data too. So you are in the range of around 20% of relative standard deviation of high numbers, which I think is very good at this stage. So here in the end of this presentation, I want to show you some really cool data that we have. So now I showed you on very high numbers, but now if we go to one patient, and then we use the protocol with the Covaris technology, and how we haven't, I should say that, we've been struggling a lot we tried actually what is available commercially on the field for solubilization of FFPE study material. Now, it's a wide window scale because in some of the patients you have very, very high stromal content, and the other ones you have not. So it's very heterogeneous too. So you really need a solution where any type of samples can be handled and processed in a good way. So let me show you data on patient, we call him patient alpha. So he's a 56-year-old male. He got colon cancer. That's when we met him, actually, half a year later. And he's still alive. And here is his history in this schematic illustration. So on the top, he got the tumor, and then he has surgical isolation of that, and they were quite many tumors within the colon. And then he proceeded, then he got treatment, then he was tumor-free, then he got recurrence, and then he got prostate cancer, and then another occurrence. So you see, these are 4 events that this patient goes through. So what you need to do in order to be able to come up with some idea of how to solve the problem, you have to do evolutionary studies, and this is an example of that. And I believe that this is the first, actually, that anybody has done on functional level, or proteomics or functional genomics, whatever you want to call it. And what happened here is that the primary first, second, and third metastases were performed in different hospitals. Switzerland, and then in Frankfurt, Germany, and then in Budapest, and then also in London. So we picked all of them up, took us 6 months, and then we moved from there. We did a in-depth characterization by pathology, so we knew exactly where we're standing, and we were following the path, and we ran through all of them from the metastasis stages as well. And Istvan, Livia, and Marcel were the pathology drivers here, Marcel from Dublin. So what you find here is as a readout where you actually can see that there's a heavy mitochondrial activation that drives the metastasis, and also a very strong push in lowering the immune defense. So the tumor is doing everything to take over. And you will find activated pathways that we were kind of figuring on how best to be able to address in terms of treatment. And also then in the third metastasis, we also saw there is a shift. You know, the primary is not the same disease as in the third metastasis. And here was a TGF-beta activation. So in the end of the day, what we found is we were able to come up with an idea of how to be able to solve. So what you see here is really cool. So the lower part of the red in there, that's a really, really very chewy, very stagnant. It's like actually glycoproteins, and it forms a barrier. It's like a mountain. So that means that immune cells and drugs cannot penetrate. It's like a barrier. That means that the tumor can grow. And that's the way how the system builds in order to be able to say. So anyhow, we came to a conclusion where we could address, and we found a combinational treatment of how you can solubilize these glycoprotein barriers. They can be small, they can be mid-sized, and they can be really large, so they aggregate. So in summary, I would say that we have been able to develop over the last year a workflow, so it will be shortly available in publication. But these are the steps that you do. in deparaphasization, you get, cross-linking you have to get rid of, and then homogenization, which is very important. So that is the part that you run it. But also up front, it's very important for us now to get an understanding of morphology. And these are the 4 steps that we do in morphology. So you take a holistic view first, then you segment it, you annotate the cells, and then from there you can actually use your AI-driven models in order to help you to provide directions of where to go. And the 4th step is really to come up with a way forward for optimal treatment of the patient. Very often what we find is combinational treatments, actually. Okay, so that was it. And of course, the most important is the people. So we have a lot of scientists, some are older, some are younger, fresh blood in, students, postdocs from 8 countries. We like Latino scientists, it's great fun. And we collaborate with many companies also. Covaris is one of them that we really like. So they provided the solution that we needed, actually, and we were able to prove that we can move forward. Okay, with that, I thank you for your attention.